All right, well, I'm really excited to chat with y'all today, and thank you so much for the invitation. I've been following the work of all of y'all for the last couple of years, so I'm excited to share a little bit about just a small slice of some of the dissertation work I did, and then also hopefully can chat more with anyone interested, so maybe future collaborations. Also, as an aside, for my postdoc work, I'm actually working on plant fungal pathogen dynamics. So if anyone's interested in plants, I'd love to chat with you all about that. But today we'll kind of focus on vectors. And then so first, I would be remiss if I didn't initially kind of acknowledge all of my funding sources and all of the help I had throughout my PhD, particularly with my advisor, Nicole Gottdinger, which I know several of you know her, and then also our collaborators in Panama. So for a big part of my dissertation work, I was really interested in understanding what can influence a vector's ability to transmit infectious diseases. So just thinking about what shapes vector-borne diseases. So we have your pathogen, the vector, the host, and the environment. And so I'm interested in looking at the pathogen, the vector, and I'm an ecologist first, and then eventually got into infectious diseases. So I'm also very interested in how changes to the environment has an impact on transmission, as well as the pathogen and the vector. So what role do microorganisms play in vectors? So microbes are important in a lot of different processes in animals, but I'll just focus on two today. So first we have development. So some old important papers in insect physiology looked at triatamines and their development. And they found that if you completely clear the gut symbionts from triatamines, they no longer develop into their adult stage. And then also pathogen resistance. So Karamotic et al. In 2011 wrote a really nice review and highlighted the capacity of gut bacteria to decrease viral and parasitic infections in mosquitoes and tsetse flies by activating their immune responses. So now for my work, I will focus on Chagas disease. So up to 15 million people in the Americas are infected. So Chagas disease is vectored by triatamines, also known as kissing bugs, and Chagas is caused by protozoan parasite Trypanosoma cruzi. So importantly, triatamines are obligate blood feeders, and so they will feed on any type of vertebrate. However, only mammals are competent hosts. And then also, interestingly, triatamines, unlike other vectors, they do not transmit the parasite via saliva. So they will take a blood meal, but then they will defecate infected feces. And then those feces get into like an open wound, particularly the bite wound, and that is how transmission occurs. So we worked in Panama, and then so the primary vector of Chagas in Panama is Rhodnius pallescens. They are found in the crowns of royal palm trees, Adelea beaudieaceae, and then in Panama, there's a quickly changing landscape, particular deforestation. And then Gottdinger et al. In 2011 and 2012 found that deforestation increased both R. pallescens abundance as well as T. cruzi infection rates. So my main questions for this kind of small slice of my dissertation is land use type associated with gut microbial diversity? And is trypanosome infection also associated with gut microbial diversity? So for our field work, so we sampled palms throughout central Panama. So we looked at pasture palms and paradomestic palms in 2 sites around the Panama Canal, La Chorrera and Capita. And then we also looked at a site in Veraguas, and there we sampled pasture palms, paradomestic palms, and forest remnants as well. And then so overall, we collected 288 samples of R. pallescens. Myself and a couple very patient, lovely undergrads dissected the mid and hind guts. We extracted the DNA, sent it off for 16S metabarcoding, and then tested each sample for T. cruzi. And then ultimately, 45% of our samples were infected with T. cruzi. So first, we're just going to look at bacterial composition. So this is looking at the top 10 genera and then relative abundance. And I think this is really striking, but we found differences in sampling regions. So Capita and La Chorrera, those 2 bars on the left, those are our 2 sites around the Panama Canal. And you can see just visually very different in the overall composition than the samples we found at Veraguas. And then so now just looking at alpha diversity, so that's within sample diversity, and we again see differences across sampling region. So there are differences across all 3, And then, but most striking is that Veraguas showed the greatest alpha diversity compared to the other two regions. And again, Veraguas is our site that was quite a bit away from the canal. And then also, so if you just kind of zoom in and only look at pasture palms, so maybe there's some differences between the palm type. So if we're only looking at pasture palms, again, across our 3 sampling sites, we are still saying that Veraguas is displayed the greatest alpha diversity compared to our other 2 sites. And then so now let's just hone in on Veraguas. So maybe there's something about Veraguas. So here we can look at habitat type. So we are saying is that our samples from the forest remnants displayed the greatest diversity compared to our other pasture and paradomestic palms, which actually show no difference. So now let's think about infection. So here, this is just looking at, so irrespective of location, just looking at the relative abundance of negative and positive samples. And here you can see that there is a difference in the overall composition of samples that are infected and samples that are uninfected with T. cruzi. So now just looking again at alpha diversity, and we are saying is that T. cruzi infected samples displayed greater alpha diversity compared to samples that were not infected. So we kind of take a step back and then look across habitat type. So what's kind of maybe driving alpha diversity? So right here, you can see that we're only looking at Veraguas and we're looking across habitat type. You're still seeing the same pattern of are displaying a greater overall diversity than our uninfected samples, kind of a little bit irrespective of habitat type. So here, just looking at very, very simple supplementary mixed models. So this is something that we need to work on further, but just initially just looked at so region, habitat type, and infection status. And we found that region and infection status were significantly driving the differences we're seeing. However, this is only explaining about 24% of the variation. So there's a lot more that we need to look at. And of course, there's no interaction effects here that we're showing. So this just leaves a lot of room for some further research. So just to kind of take a step back and look at everything. So we're seeing regional differences across our samples. We're seeing that there's greater diversity at forest remnants. We're seeing that T. cruzi infection is associated with an increase in bacterial diversity. And that sampling region and infection are driving differences in diversity, but likely is influenced by a habitat type. Again, we're seeing some differences in forest remnants versus our pasture and paradomestic palms. So just some future directions. So I've kind of set my PhD work aside just a little bit, but as I'm kind of sort of transitioning to my postdoc, but I have so much other data and a lot more things to look at. So I'm also happy to chat with folks who have any thoughts. But so further, we have blood meal data. So I didn't have a chance to talk about this, and there's a lot more I need to analyze. But we were able to identify 2 genus of all the different blood meals that the kissing bugs fed on. So really looking at just host diversity from our forest palms or pasture paradomestic palms, as well as our different sites in our different areas, and as well as kind of relationship with infection. And then also looking at differential abundance. So really that just means tracking specific microbes and seeing if there's greater or lower abundance depending on statuses of infection or location where we're finding them. And then ultimately, I think such a big question that lots of microbiome folks think about is, okay, so we're seeing differences in diversity. Does that really matter? What is the functional diversity? And really, can we, maybe make some hypotheses and some interpretations to decide if there's any risk of higher susceptibility, just depending on the different microbes present, or if they're all kind of just doing the same thing, just at different rates. So that is all I have for you today. I kind of lost track of time, so hopefully I didn't speak too long, but thank you again for listening. I would love to chat with anyone further. You can contact me with my email. Twitter is kind of a dying social media, but I'm still on there. I still check my DM. So again, I would love to chat with folks further. Thank you.